Hello students, welcome to uttyoron. Now in the previous lecture we have talked about the kingdom Protista, which comes under the chapter biological classification. Now this will be the first video of the series on kingdom fungi. So today we will be starting with the preliminaries of the kingdom fungi. So first let us talk about the general characteristics. So this is a typical structure of fungi. Now fungi are very common example, a very common type of fungi which you have come across would be the bread mold. It has a scientific name that is Rhizopus. It's a very typical fungi and let us see the parts of the fungi. So here you can see it is the hyphae. These are small, thin filament-like structure which crawls over the substratum. Now it needs a certain level of anchoring in order to absorb the nutrients from the substratum, as it is a saprophyte or parasite. We already know that. Why? In kingdom Protista, we had slime molds, which are the assumed predecessors, ancestors of the kingdom fungi. So here also the fungi are heterotrophic and they exhibit saprophytic or parasitic mode of nutrition. So this hyphae, these are thin filaments which give rise to root-like structures called rhizoids. Here you can see sporangiophore, sporangium. So sporangium is the structure which bears the spores. So this full thing is called sporangiophore, and the bulb-like structures that you see on the top is the sporangium which contains the spores. Now spores are the reproductive structures. They can be sexual or asexual depending on the organism. So they are means of propagation for the organism. So when these spores, you can see over here, some spores are released when the sporangium bursts. So these spores, once they find a suitable substratum, which contains enough nutrients and also the weather is also favorable for them to germinate, they germinate and produce the vegetative body. So these are the general characteristics, a few notable characteristics. One will be they are heterotroph. They heterotroph. They favor moist places. Most of the fungi you will find, they favor moist places. All right. And they have this typical structure. Some of them may be unicellular. Now, I will be discussing in details about the body plan over here, which is prevalent throughout the fungal kingdom. This is sort of a summary of the body plan. See, there are some fungi which has reduced thallus, that means very small thallus, inconspicuous thallus. So under that, we have the unicellular ones, that is the yeast. We have filamentous, dimorphic, and we have the amoeboid or plasmodial forms. So these have reduced thallus. Now moving to a bit advanced ones, which have a little bit of prominent thallus, hypha and mycelium. They can be septate or aseptate. What are septate hypha? Suppose this is a hyphae, a portion of the hyphae. It may have partitions inside it. That is called septum. When it is present, it is called septate hyphae, and when the hyphae does not have septum, it is called sinusitic or aseptate also. Aseptate hyphae. Next, a bit more advanced forms, they show tissue differentiation and division of labor. Not as much as the ones that we see in the higher forms like plants and animals. Whenever we speak of tissue differentiation, we have this tendency of imagining the complex one. They show minimum, the fungi, they show minimum tissue differentiation. In somatic and reproductive structures, we have the rhizomorphs, stroma, and sclerotia. These are a few special structures that we will discuss later on. And then in the tissue form, we have plectenthyma, We have prosenchyma and we have pseudoparenchyma. So the difference between prosenchyma and pseudoparenchyma is that in case of prosenchyma, the hyphae are loosely interbound with each other. So you will see that the mass is tangled but not so much, so that it gives a tissue-like appearance. But in case of pseudoparenchyma, we already know what are parenchymatous cells, right? Those are the storage cells, the simple cells that we, simple tissue. It comes under the simple tissue of the chapter plant tissue. You've heard about it. So pseudoparenchyma means they almost appear like parenchyma. They are not parenchyma. They are made up of fungal hyphae. But the interconnection is so compact, they are so compactly arranged, they give this appearance of parenchyma. So this is the general body plan that is existing in the kingdom fungi. Now let us talk about the reproduction process in general for fungi. We know that they produce spores. So spores, when they germinate, whenever you are learning a reproductive cycle, you have to choose a starting point. So let's take the spores as the starting point. Now from the spores, when they are germinating, it is producing the mycelium. Now this mycelium, it enters the reproductive cycle. It produces and it goes into a heterokaryotic stage. That means two nucleus having different properties are there to demonstrate plasmogamy. What is plasmogamy? Haploid cells from two different mycelia fuse to form a heterokaryotic cell. In fungi, you will see sexuality. They exhibit sexuality, not in the form as we are habituated in higher organisms like plants and animals, but they exhibit sexuality. One will be of positive nature that we consider as the male, another mycelium will be of negative nature that we consider as the female. So two different mycelia on the basis of sexuality, they fuse to form a heterokaryotic cell with two or more nucleus. So this is the heterokaryotic stage. Then we have karyogamy. What does the name suggest? Karyo means nucleus, gamy means fusion. So the nucleus fused to form a diploid zygote. The zygote is 2N. Then this zygote undergoes meiosis. Why? The vegetative structure was N. See, it had the chromosome configuration of N. So after meiosis, it gives rise to the spores, which on achieving the favorable conditions and substratum germinates to form mycelium. So this completes the sexual reproduction. Now, how will this mycelium reproduce by asexual reproduction? This mycelium, if it wants to propagate by asexual reproduction, it will just undergo mitosis. It is undergoing mitosis, remember, not meiosis, because it is already haploid. It is forming more haploid bodies with the formation of mitospores. Okay, there are two types of spores, meiospores and mitospores. As the name suggests, these spores are formed on the basis of the process that is behind the formation. If it is formed by mitosis, it is mitospore. If it is formed by meiosis, it is meiospores. So here I have uh, accumulated the different ways of reproduction observed in the kingdom fungi. So they reproduce both by asexual and sexual method. What are the asexual means? Spore formation. Just now I have discussed what are spores. So they reproduce by spore formation. Next, they have conidia formation. Conidia are special structures that are formed at the top of the hyphae, which when separated and on receive, they behave just like spores. When these cells are, except they are not spores, they are cells. When the cells they separate, you can compare conidia formation with the fragmentation process. Except here, the conidia are separately formed. In fragmentation, we have learned that the organism simply breaks and regenerates. But conidia is a special type of cell meant as a mode of, meant for participating in asexual reproduction. So this conidia, when it receives a favorable condition, it behaves just like spores and germinates. Next, under sexual reproduction, you will see isogamy, anisogamy, and polygamy. In the previous videos, I have discussed in details about these three types of sexual reproduction. So these three are observed in the kingdom fungi. Now let us move on to the classification of fungi. The kingdom fungi can be divided into two prominent parts, Oomycota and Eumycota. So what is Oomycota? Mycelium aseptate. Okay, mycelium is aseptate. It does not have septa. Whereas Eumycota is also known as true fungi. They are the higher forms of fungi and they exhibit septate mycelium. Under Oomycetes, under Oomycota, we have, sorry, not Oomycetes, under Oomycota, we have Phycomycetes, which is divided into two groups, Oomycetes and Zygomycetes. So this Oomycetes is often considered under the group of algae as they are more, they are not considered under the group of algae, but they exhibit characters which are more inclined towards the algal groups. They do not exhibit characters, they do not behave as much as like the fungi, they behave more like algae. And Zygomycetes, which is also known as conjugation fungi. Under Eumycota, we have the three groups, Ascomycetes, Basidiomycetes, Deuteromycetes. Ascomycetes is sac fungi. Why? The spore structure that they produce is known as ascus. So from ascus, the name comes Ascomycota, and ascus means sac, so sac fungi. Basidiomycota, they give rise to basidium, which is the club, which is club shaped, hence club fungi. And Deuteromycetes, they are fungi imperfecti. Why? Sexual reproduction is not observed in the Deuteromycetes group. Okay, so let's start with the first group, that is Oomycetes. Now, Oomycetes are also called algal fungi or water moons. Just now I have discussed they are uh, extremely attracted to water, and they are more close in the characters to the algae than they are to fungi. Hence, they are called water molds. Why water molds? They are a bit like fungi. Like we had slime molds. As the name suggests, molds means they are a little bit closer to fungi. But here, uh, water mold means they behave just like algae. Next, they include one of the devastating plant pathogens, Phytophthora group belongs to Oomycetes. They cause innumerable numbers of, not innumerable, they cause many plant diseases. Some of the diseases caused are late blight of potato, downy mildew of grapevine, root and stem rot of soybean. So this picture over here, it is also a species of Phytophthora. It has this peculiarity of having this lemon-shaped sporangium. Next, hyphae are multinucleate and aseptate. This is a characteristic of Oomycetes. Why? They belong to the uh, division Oomycota, which is basically the aseptate fungi. Next, vegetative nucleus is typically diploid. Cell wall is made up of beta-1,3 glucan and beta-1,6 glucans. Okay, so as we know, we have this knowledge so far that cell wall of fungi is made up of chitin, but Oomycetes, the cell wall is mostly made up of beta-1,3 glucan and beta-1,6 glucan. Chitin is found in minimum number of, very few uh, species have minute amounts of chitin in their cell wall. So the dominant composition is beta-1,3 and beta-1,6 glucans. Next, asexual reproduction is by the formation of motile zoospores. The zoospores also exhibit a typical characteristics, that is, they are biflagellate. That means bi means two. They have two flagella. One is present at the posterior end, another is present at the anterior end. And this anterior flagella is tinsel type. That means it has hairs. And the posterior flagella is of whiplash type. Heterothallic mostly. So the group Oomycetes are mostly heterothallic. Now I told you fungi exhibits sexuality, right? So heterothallus means it's similar to heterosexual, the term we use for higher organisms. So heterothallus is same. There are two thalli, each of different nature. So they are mostly heterothallic except Pythian and some species of Phytophthora. Separate thallus produces antheridia and oogonia. So this antheridium is the male or positive and the oogonia is the female or negative. Okay. Paragynous. What is the paragynous condition? When antheridia is present at the side of the oogonia. And amphigynous condition is when the antheridia surrounds the base of the oogonia. In the previous slide, I have mentioned about a disease, sudden, this one, a disease caused in the oak trees. So here you can see what the condition is of the plant. The fungi has literally damaged the bark of the tree. So this is a pretty devastating group if you analyze. So this is all about the group Oomycetes. Now let us move to the next group, that is Zygomycetes. Now this is the group which you come across very popularly, very frequently. Zygomycetes, over here you can see this is Rhizopus or bread mold. This is the sporangium. This is the sporangiophore. Inside there are spores. This is a remnant of a burst sporangium. And here these are the mycelial mass. So they are mostly terrestrial. The hyphae is cenocytic, that means multinucleate and aseptate, known as conjugative fungi. Okay, they reproduce by gametalgingeal copulation and they exhibit conjugation and hence they are called conjugative fungi. Next, prominent asexual spores called sporangiospores are present in this group, Zygomycetes. Some forms they exhibit chlamydospores also. As I mentioned just now, they reproduce by gametangial copulation and hence they are called conjugative fungi. Now, what is gametangial copulation? What happens? Uh, I will rather dedicate a class on the different modes of reproduction because it is pretty much basic. There are at least two, three types. There are two most similar words, gametangial copulation and gametangial contact. These are two processes found in fungi for sexual reproduction. Now, the term is very similar. The subtle difference is that in case of copulation, the gametangium that is formed, it is not retained. Whereas in case of gametangial contact, a tube is formed through which the nuclei is transferred. So in details, I'll be discussing the reproduction later on in the video. So that's all for today class. Please stay connected with Uttyoron on different social media handles and do revise your concepts from different sources. I would rather suggest that you stick to any one source because as you combine different sources, you will see that you are getting confused. One source is enough. If you stick to it, you will see that the most of the syllabus is covered in the easy way. So please stay connected with us. Thank you.